Record, start record. Well, good morning, everyone. This is our EM-X starting 2026. I've had a lot of greetings about hoping that 2026 is better than 2025. As you know, it hasn't gone off to a greater start right now, but we are all scientists, all electron microscopists, and so we tend to work internationally at our field. So we have another exciting group of talks coming up and starting today with the bio talk and followed by the material science talk. Of course, this year has the Football World Cup and the World Cup of Electron Microscopy in Liverpool, which is my hometown. So I hope to see everyone there. The deadline for submission is March 2nd, which is also the next EM-X for us. So without further ado, I'll introduce Wah, who will make his own greetings and introduce our biological speaker. Please, Wah.
Okay, good morning, everyone. It's a great morning here in Stanford. The sun is very shiny, the weather is warm, and so it's my great pleasure to introduce Danev from University of Tokyo. I know Rado when he was a graduate student where he developed the techniques for phase space in the graduate school in Japan. And then he's the first one to make the so-called Zernike phase space working for biological sample. That really surprised the world how wonderful the image contrast was. And after he finished his PhD with Professor Nakayama in Japan, he moved to Munich in the Max Planck Institute for Biochemistry in Martin Street, where he developed the Volta phase space, which is a kind of commercial product now in Thermo Fisher. Again, it also make the phase space much easier to operate than the Zernike phase space. And not long ago, he moved to Japan and is now the professor in University of Tokyo. And today he's going to tell us another technology exploration he has done that using 200 kilovolt electron microscope for high resolution cryo-EM studies. Okay, Rado, you're very welcome to speak to us.
Thanks, Wah, and thanks to the team that organizes the seminar series for this opportunity to present our work. So let me see. The screen should be there now. Let me know if there is a problem. So yeah, I guess this will be a contribution to the World Cup of Electron Microscopy as mentioned earlier. So we did some performance testing on a 200 kV electron microscope that was introduced recently with some new upgraded hardware, and we were able to get some atomic resolution results. So starting a bit from afar, the cryo-EM resolution revolution technically started around 2011 when the direct electron detectors were introduced. But as you can see here from the early PDB depositions, it kind of took off around 2015-16. So roughly 10 years ago, we can say that the real cryo-EM resolution revolution started. If we plot the cryo-EM depositions on top of the X-ray depositions, we can see that we are still haven't overcome the X-ray in terms of number of depositions, but it's getting there. And if we plot just the membrane protein structures at resolutions better than 5 angstroms, you can see here that cryo-EM is definitely contributing a lot more structures nowadays. And it seems that groups that have used X-ray in the past, maybe for membrane protein studies, are either switching or just transitioning to using cryo-EM for these structures. Now, these kind of structures are done on the kinds of microscopes you see here. So we have the two main manufacturers, Thermo Fisher Scientific with their Cryos 5 microscope. It has a nice box and also colorful light that changes in the front. And then the GOL, which is the Japanese manufacturer, has the cryo-EM 300 version 2. That is also in a box. It's a bit bigger and also looks neat from the outside. On the inside of these boxes, of course, we have the typical microscope columns with all the wires and so on. Now, the second-tier microscopes from both manufacturers operate at 200 kilovolts. Here you see on the left the Thermo Fisher Glacius 2. It's an upgrade of their Glacius microscope with the cold field emission gun, and maybe some optimization in terms of housing the hardware in a single box and maybe less ancillary equipment on the side. And then from GOL, we have the cryo-EM 200 version 2, which I'm going to talk about later in the talk. Now, I forgot to mention, so the big machines, they cost around 9 coins. I don't know the exact price nowadays. I haven't ordered the microscope recently, but we can say 9 Mario coins, for example, for the 300 kV machines, and then around 3 coins for the 200 kV machines. So roughly 1/3 of the cost of the big machines. Now, if you look at the positions by the two types of microscopes, here you see 300 versus 200, there is a big discrepancy. So it's roughly 1:9 or 1:10 in terms of number of depositions. And this ratio hasn't changed much, apart from the last couple of years where it seems that 200 kV machines are starting to kind of gain more ground. But in general, judging by the depositions, it's 1:10, which is quite, surprising when I saw that. If you look at the membrane proteins, the discrepancy is even bigger, so it's around 1:15, if I remember correctly. So still 200 kV machines seem not to be appreciated too much. Now, one would wonder, why is this discrepancy? Could it be due to resolution differences? Here you see statistics on the deposited structures, deposited maps in the electron microscopy data bank in the last 6 years. So we have the two histograms, and they have been normalized because the total number of structures, as you can see here in the boxes, is roughly 1:9. So the histograms are normalized, and then we also fit those with the log normal distributions. They fit very nicely, and surprisingly, the difference in resolution is just 0.25 angstrom. So it's a quarter of an angstrom difference, but as you saw, the number of structures difference is so huge. So the devil here is also in the detail, and if you look at the high resolution region below 3 angstrom, then we see that the discrepancy is getting bigger. So for example, the region between 2.5 and 3 angstrom, the 300 kV is about 2 times more prolific, relatively speaking, because these are normalized histograms. And then in the region below 2.5 angstrom, it's 3 times more prolific than the 200 kV. So in these regions, actually, I have this kind of mark. So in these regions is where we get, good quality maps that help us get higher fidelity models, better modeling of side chains, also small molecules, if we have some kind of a ligand, and water molecules, which are becoming more important for actually evaluating and investigating the function of proteins. Now, Gabe Lander's group was one of the pioneers that did a lot of work in pushing the performance of 200 kV machines in the past. It's almost 10 years ago, you see here. So they published some amazing structures. Here on the left, you see the rabbit aldolase at 2.6 angstrom. At the time, that was amazing result. And then a few years later, they published also apofertin at 1.75 angstrom. So this is, they called it a Team 200. So basically people who really believed in the 200 kV machines and their capabilities. And I'm now kind of, I would like to join this team, as you would see later. And I also am a strong believer of the capabilities of kind of more efficient or cost-efficient machines. Here you see more recent results from the 200 kV machine. So we have here the CDK activating kinase at 1.7 angstrom. This is an amazing result also from collaboration, I guess, between Abhay Kotecha and Basio Graber. So they did multiple structures, more than, I think, around 15 structures of this protein with different inhibitors. And then by combining the data sets, they were able to get sub-2 angstrom, kind of deep sub-2 angstrom resolution of this protein. And then the most the highest resolution 200 kV structure of apofertin I found on the EMDB is 1.5 angstrom by Abhay and Koch. I guess they haven't published it yet, but this is the highest one that's available on the EMDB. Now, the machine we used for these tests is a Geo cryo-EM 200, which is now version 2. The time when we ordered it 3 years ago, it was not yet, didn't have such a designation, so it was just a 200 kV cryo-EM, which we called it special because we requested some modifications. So the base specs are 200 kilovolts, cold field emission gun. Joe's machines are all with the cold field emission gun. It has a 12 sample autoloader and an in-column omega type energy filter. So the Joe machines, they have an energy filter as a standard. And then we requested a high resolution objective lens pole piece. It has a narrow gap and also lower spherical and chromatic aberrations as shown here. So the standard values are 2.7 for the spherical and 2.8 for the chromatic. So you see these are lower values, so better optical performance. We also asked Joe to remove the objective mini lens. This is a lens which they traditionally had kind of in the bore of the objective lens and was used for low magnification observation, but it caused a lot of thermal drift. So switching between low mag and high mag caused a lot of drift. And by removing this lens, they optimized the overall performance. Also, the vacuum system was simplified, so smaller and quieter pumps and requiring less service. And finally, electrostatic dose modulator, which is a very fast electrostatic shutter, which can be used both as electrostatic shutter and as a beam intensity modulator by applying, modulating frequency, which is much higher than the frame rate of the detector. So the basic optical performance of the microscope is determined by the contrast transfer function temporal coherence envelope. And here you see in this plot the temporal coherence envelope simulated for a number of microscope configurations. So starting with the Joe cryo-EM 302 and Krios 5, they are both cold field emission microscopes with the standard objective lens pole pieces. This is the green curve. Now, there is a new cryo-EM 300 version 2 with a narrow gap objective lens pole piece that was recently installed in Okinawa at the OIST. I haven't put the curve here, but my guess is that it will be even higher than the green line here. Then the cryo-EM 200 version 2, which I'm presenting about here, is the purple line, so it comes in second. Then we have the thermovisual glaciers 2 here, the blue line. Then comes the traditional Krios with the Schottky FEG, here the dashed green line, and other Schottky FEG machines. So you see here the cold field emission gun provides a kind of a big boost in the overall optical performance on its own. Of course, when combined with the narrow gap objective lens pole piece, you see also it gives another push. Yeah, so I just kind of limited here the range that is most interesting to us, at least, I would guess, in terms of structural biology nowadays. So between the 2 and 3 angstrom range, you can see here that, of course, the 300 and the 200 version 2 are top, and then comes the Glaciers 2 and the other machines. So if you want optimal performance in this range, you have to consider this kind of comparison here. Now, to test the machine initially, we used oriented single crystal gold sample. Took some images, and here you see the Fourier transform of one of the best images. So it shows spots along the tilt axis. So the tilt axis is something like this. I don't have it marked here because I don't know actually exactly where is the tilt axis. So along the tilt axis, we see spots up to 0.656 angstrom resolution, and perpendicular to the tilt axis, they go up to 0.7. So in all direction, the resolution goes beyond 1 angstrom, which is I think very satisfactory for cryo-EM applications where we don't reach close to 1 angstrom usually. Then to further test the machine, we collected apophotine data sets, and here is the one that was actually recently, we submitted a preprint last week, which reached atomic resolution. So the configuration for this sample was using 0.6 micron whole ultra AU foil grid. So this improved the stability of the sample. We are also collecting a single image in the center of each hole and having the beam uniformly illuminating the hole all around. So this was to minimize beam-induced specimen motion. Here you see an example image from the data set. Of course, the apophotine is nicely visible as spherical molecules. Then after reconstruction, this is what the map looks like, and the data reached 1.24 angstrom, which we call, according to the current consensus, atomic resolution. Then Yamashita-san from University of Tokyo, he's actually the creator of the ServoCAD software. He was very kind to help with the modeling of this map. So here you see a representative side chains densities and also the model. So the gray surface shows the map at the lower threshold, the typical one which is used to show the map. Then at a higher threshold, we have the blue surface, where you can see individual blobs for the non-hydrogen atoms in most of the side chains. And then by calculating a difference map between the hydrogen atom omitted model simulated map and the experimental map, he created the difference map, which is shown in green. And at the normalized 3 sigma level, we can see densities, putative densities for hydrogen atoms at most of the positions in the side chains that are shown here. So this is a very nice result also in terms of the identification and location of hydrogen densities. To further quantify the performance, we also calculated a Rosenthal-Henderson plot. This is done by taking random subsets from the original data sets and then independently doing 3D refinement on them and then plotting the resulting resolution in a plot which has the logarithm of the number of particles on the horizontal axis and 1 over the resolution square on the vertical axis. And theoretically, this should be a linear plot. And you see it's very close to linear. So we did linear fits through these data points. And from the slope of this fit, we can actually measure the B factor. So for the cryo-200 version 2, the B factor was 39 angstrom squared. And the second data set we evaluated was from our Cryos version 4, Cryos G4, which has a Schottky FEG. So it's not a cold FEG. With this one, we got also a B factor at 38.7, so virtually identical to the one of the cryo-200 version 2. Now you see that the curves are a bit offset vertically, and this would mean that the overall signal-to-noise ratio in the data for all the spatial frequency is a bit higher for the cryo-200. Basically, it needs a bit less particles to reach a given resolution than the Cryos dataset. Now, I was also curious about the effect of radiation damage on the achievable resolution. So to simulate that, I took the first 1,000 movies with around 58,000 particles and then did independent movie alignment of these by omitting varying numbers of initial frames. This is to simulate basically pre-exposure or the sample being pre-damaged, so to say. And then doing just 3D refinement, no polishing, no other steps. This is the resulting data. So here you see on horizontal axis the pre-exposure to the sample. And then the resolution is on the vertical one, so the red dots represent the results. And it was this data was fit very well by just a parabola. I also did some theoretical estimation of what the resolution would be based on an empirical damage model by Nico Grigoriev and Tim Grant. And the black points here, this is the simulation using the same exposure that was measured in the experiment. And by adjusting the exposure, reducing it to 82% of the experimental exposure, it got much closer to the simulated one. So I'm not sure if the pre-exposure is, the measured exposure is not accurate, or the empirical model is a little bit off. Now, what is interesting here is that the model, the damage starts from the beginning. So basically the next point is already at a substantially or kind of noticeably lower resolution compared to what we see in our experiment. So by removing the first 5 frames, we didn't see a big change in resolution, which would mean that maybe there is still some motion there that affects the initial frame. So they are not fully contributing their information to the reconstruction. So that's something to consider in practice. These are examples of map features at the different pre-exposure levels. So we see, of course, degradation in the overall fidelity of the map. And what was surprising to me, at least, is that even after about 20 angstrom of pre-exposure, 20 electrons, per square angstrom of pre-exposure, the map still reached below 3 angstrom. So, of course, the water densities are completely gone by then. So these are the water molecules here. And you see by that time, of course, they are completely gone, but still under 3 angstrom after 20 electron per square angstrom was a bit kind of surprising. So I also collected quite a few GPCR datasets, G protein coupled receptor datasets. So this is a real world sample from our collaboration with the metabolic GPCR biology lab at the Monash University. So here you see the reconstructions. Overall, they correspond pretty closely to what we have been getting from the Cruise G4. So this is the Schottky effect Cruise, which I showed in the apophethine comparison plot. And similar to what we got from the Cruise G4, we also, one of the GPCRs actually reached also sub 2 angstrom resolution, which is, I haven't checked the PDB recently, but it was a few months ago, it was still only the third G protein capote receptor structure below 2 angstrom, the other two being done on the 300 kV Cruise G4. So this overall, the impression is that the results from the Cryo-EM 202 are similar, very similar in performance on the outcome to the results from the Cruise G4, a 300 kV machine. Now, another practical, actually real-world application was in collaboration with Jian from Kikawa's lab. So he's working on the structural studies of kinesin, in this case kinesin 2, and it was a heterotrimeric complex of the kinesin together with the adapter, and then also with the adapter and the cargo. So we have two structures. One is without the cargo and the other one is with the cargo. So this is the first time such thing to be visualized or to be structurally observed. The structure without the cargo was behaving much better, so we had more orientations and the structure was isotropic, or nicely isotropic, let's say. The one with the cargo suffered from very strong preferred orientation, as you can see here. So we have streaking, but Jian was still able to build a decent model out of this. Now, another very interesting thing to investigate is going to even lower voltage. So 100 kV, that's kind of the promise is that these kind of voltages will provide sufficient performance for getting, decent initial data sets, let's say, and also for sample screening, while the machine and the maintenance will cost much substantially less than the big machines, 200 and 300 kV. So for this, I contacted Dectris, and they were kind enough to give us, to loan us their Dectris singular camera, which you see here. It's a high-speed hybrid pixel detector. That means that each pixel has its counter, its own counter. So when when an electron hits the pixel, then the pixel itself is doing the counting, and this can provide very, very high speed counting speeds, as you can see here, 10 to the 7 electrons per second per pixel. Of course, the detector has a very large pixel to be able to do that. You see here 75 by 75 microns. That's 15. I think I'm not sure if I can calculate it this hour, but 15 times larger than the typical direct detector now in electron microscopy. Also, the chip itself is much larger. So when it's mounted on the microscope, it's also much larger area. And it's composed of two modules. You can see them here, and there is a gap between the modules. So this is a sample image from the apophethine test data sets at 100 kV. You see here we had to kind of compromise between pixel size and number of particles. So we used 1.17 angstrom pixel. The magnification we used was very high, quite a bit above the typical magnifications used in cryo-EM, so 500,000. That's a range which typically is not used on cryo-EMs in general. So it was a bit of a challenge to operate at these high magnifications. You also see here the gap in the middle that is interpolated in software after the data is collected and is being processed. And another thing is that in cryo-EM, when a particle is close to the edge of the image, it typically is not usable because part of the information is delocalized due to defocus outside of the image. So you can see from this example image that not very many particles will be usable from this kind of image. So hopefully future generations of these detectors will be gapless in the middle, so that at least the central portion is not sacrificed. This dataset reached 1.9 angstrom resolution, 1.91, and with 250,000 particles. I was a bit kind of more optimistic when I was collecting the data. At least the data looked shiny or crisper. But of course, considering the larger pixel, that's, I guess, to be expected. So the dataset actually reached beyond Nyquist, of course, but 23% beyond the physical Nyquist, so 123%. The practical challenges, so one that I mentioned already is using very high magnification, so that was very unusual. and calibrating the microscope at this magnification was also not very easy. It was not fully calibrated, but already the time for the loan was coming close to the end. So anyways, it was not an optimal setting. So there may have been some, ways to improve it further, I guess, if we had more time. Now, there are also recent results also at 100 keV by Gabe Lander and Clement Verba. So they did tests of the Gatan Alpine detector, which is also designed to operate at the lower voltages, 100 and 200 keV. And they got very similar resolution from their dataset as well. So these are the two datasets are very close to each other. And they show that in principle, it's possible to get, sub 2 angstrom resolution results at 100 keV, but it's challenging. I mean, the personal experience is, it's more challenging than getting them on the 200 or 300 keV microscopes. Now, another interesting test that I decided to do is to collect a data set of myoglobin. Myoglobin is just 17 kilodalton, which is nowadays, I think, according to some of the theoretical estimations of what the cryo-EM limit is, it's on the limit. So 17 kilodaltons is considered the theoretical limit. And in any case, we should try, right? So here on the right, on the left, you see a data set which was collected, again, almost 10 years ago by Mariam Koshue. She was a student in my group at the Max Planck Institute, and at the time we were working on the Volta faceplate. So this data set was done with the Volta faceplate on a Krios, and she got some kind of promising 2D classes. As you can see here, some of them look like some of the views of the model of myoglobin. Now on the cryo at 100 keV with the Dectris Singla, I basically got the same result. Of course, the outlook of the data is different. I haven't tried processing this yet with Reliant. I would do that because you see these classes from Reliant, they look smoother, not so much overfitting as the classes from these are done on CryoSpark. But the classes that show some promise are very similar in both datasets. So you see here the side view class. I have also here on the bottom simulated map projections from the model. So these kind of classes where we see more contrast features, they will be more viable to achieve in practice, of course. And orientations of the molecule where you have less features and less contrast, of course, these kind of classes will be more difficult to get. And also these kind of orientations in the tree reconstruction will also be more challenging to assign. Therefore, I think we are kind of on the verge, you can say. So we get some classes that show features that are similar to the projected ones, but still no outcome in 3D. Now, if you look at the temporal envelopes at the 100 keV for different configurations, one thing is shining here, and it is the so-called Dublin lens. This is a new lens that was designed in collaboration between the MRC LMB in the UK. So Greg McMullen, Richard Henderson, and Chris Russo, and Lewis Jones. So they designed the lens that has a chromatic aberration coefficient of just 1 millimeter. So this is the lowest chromatic aberration lens I've seen so far. Maybe in material science there are better ones, but for cryo you need to have some space for the sample and also for cryo shields. In any case, if you combine this Dublin lens with maybe we can go to this one. So if you combine the Dublin lens with the CFEG, so with the cold FEG, it will provide almost uncompromised optical performance in the 3 to 2 angstrom range. Now the cryo-arm 200 with the narrow gap pole piece, it already starts to hit a bit below 2.5 angstrom, and then the other machines like the Glacius with the narrow gap. I forgot to mention the Gabe Lander's dataset here was collected on a Glacius with a narrow gap objective lens. So it's a 1.7 millimeter with the Schottky FEG though, so it's not a cold FEG like the cryo. And then we have the Glacius, and then the Dublin lens with the Schottky FEG. Here is showing purple. And on the bottom here is the Tundra, which is a standard product. It is with a narrow gap, but it has a Schottky FEG source. Now, this is a very important plot we should consider. It was calculated by Matthew Pitt and Chris Russo in their amazing work on measuring the inelastic and elastic scattering cross sections at various voltages, and from that they calculated the optimal accelerating voltage versus sample thickness. So here we have the sample thickness on the horizontal axis and voltage on the vertical axis. So at 100 keV it's about 30 nanometer. At 200 keV it's about 50 nanometer, and then at 300 keV, of course, larger than that. So 300 keV at the moment is the highest voltage we have in practice for cryo-EM experiments. Hopefully in the future there will be some kind of I guess prototypes exploring higher voltages for tomography especially, but at the moment this is what we have. But in any case, 200 keV at 50 nanometer and 100 keV at 30 nanometer, this is something we have to consider. Now looking at other factors which are also very important, not just the optical performance, it's the cost of the machines. So we have, I'm just comparing the Joe machines here, we have the 300 and the 200. Again, the cost is, the purchase price is about 3 times higher for the 300 keV. Installation space is 1.5 times larger, I guess, typically. Power consumption also about 1.5 times higher. And the service costs, which I don't know about, are, I guess, 2 times or so higher for the 300 kV. So considering all this and the difference in performance that we show with the highly optimized 200 kV machine, I think in near future we can start to see some kind of shifts in or reshaping in the cryo-EM performance landscape. So thanks to the cold FEC and the low CC, low chromatic aberration objective lenses, this could happen. And what could actually happen is handing down of roles in the across the voltages. So for example, single particle analysis project to move more of them to 200 kV high performance machines, cold FECs and narrow gap objective lenses, screening and initial data set collection from the 200 to 100 kV, and the 300 kV machines can provide more time for things like tomography and thicker samples. Of course, the ultimate goal is to make cryo-EM more accessible, affordable, and prolific in the end. So with that, I would like to thank our collaborators, so Masaki Kawa, Yanagisawa-san, Jian, and Yamashita-san from the University of Tokyo, the GPCR team at the Monash University. This has been, over the years, in the past, it has been an amazing collaboration with them. Then the people from JOL who helped with the alignment and configuration of the microscope at 100 kV, and of course the people from Dectris that helped with the loaning, installation, configuration, and operation of the camera. And thank you very much for your attention. I hope I can answer if there are any questions. And finally, yes, I kind of rushed to submit the preprint to bioRxiv, so it's now on bioRxiv. The same results you saw here, they're in this paper there. Thank you.
Okay, Rado, it's amazing work. Thanks very much for carrying out all this experiment. It's extremely exciting to see these kinds of quantitative analysis. Okay, so there are some questions that from the audience. Let me see if I can. Okay. Okay, so one of the questions is for the 200 kilovolt Apelferton data, what chances do you see when the resolution go beyond the Nyquist?
The 100 kV or 200 kV data
set? The 200 kilovolt.
The 200 didn't go beyond Nyquist. The 200 is
that's right.
Far below Nyquist. Yes.
Far below the Nyquist. So it's only the 100.
Yeah, the 100 went beyond Nyquist.
Okay. And then there's another question. Do you see any damage difference between different voltages?
I didn't quantify it, but it is to be expected. I think indeed for the 100 kV, one thing in practice was that I use kind of typical exposure, about 50 electrons. And when I was processing the dataset during the polishing step, I noticed that towards the later frames, there was kind of overfitting of the high resolution information. So when doing the dose weighting, this practical, what is that called? Data-driven dose weighting, the later frames were kind of overweighted. The high frequencies, there is a kind of an artifact which appears there, and that actually affected the performance. So what I did in the end is I just used the first 30 frames of the movies, so basically half the total exposure, and this improved the resolution to 1.9 angstrom. So I would definitely expect higher damage, as shown by the inelastic cross section here, measured by Pete and Matthew Pete and Chris Russo. Definitely there should be higher damage.
Okay. And another question is, what is your setup for the detectors at the 200 kilovolts cryo-EM?
It's a Gatan K3, a standard Gatan K3 camera.
Oh, okay. Okay. And okay, Bob, you have any questions?
Yeah, a question I had. Thanks very much. This is fascinating. I know that the LMB people are also looking at 100 kV microscopes as well. In material science, we go for aberration corrected machines now, and you didn't really talk about that at all in comparison with these cryo-EMs. So I'd say that aberration corrected is almost the norm now in material science. And do you see that coming for bio in the future, or what's your feeling about that?
So spherical aberration correction, I don't think changes anything much because we correct our aberrations post-acquisition. We can do that in processing. Of course, chromatic aberration correction, that's what the people at the LMB are excited about. So they have a paper demonstrating that there should be, if I remember correctly, almost a doubling of the signal if you do chromatic aberration correction and low defocus together. I personally, I haven't had the resources or the enthusiasm to go there. I'm sure that maybe when we push all the available, when we kind of squeeze all the available juice from the non-aberration corrected configuration, then that would be the next step probably. But I would say we are still far from maximizing the performance envelope without aberration correction. Once this is done, maybe we should move to the aberration correction.
Thank you.
Thank you. There's a question from Dr. DeRosier. Do you use defocus for achieving contrast? And how much would the performance be improved if one use laser phase plate?
Yes, of course, we use defocus. I try to use lower defocus for the upper 13 datasets, especially because you have less delocalization of the information, and you don't need such a high contrast with this size of molecule. With the laser faceplate, if you are able to collect your data perfectly in focus, which in practice is quite challenging actually. It gets even more exponentially more challenging the higher the resolution you want to achieve. Then in focus with the faceplate, you get doubling of the signal. So theoretically, with the faceplate, you should be able to, get better results if you collect the data in focus. If you collect the data in focus, with the faceplate and defocus, I don't expect big differences in terms of the ultimate performance with samples like upper between the conventional and the faceplate.
Yeah, I suppose David may refer to the hemoglobin types of sample.
For the small ones, yeah. For the something like the hemoglobin and smaller, maybe even myoglobin, that's definitely something that the faceplate people are probably already trying or they plan to try, because that's the area where the faceplate will help again by kind of boosting the low frequencies and providing you zero free CTF, basically no zeros. Doubling the signal, that would definitely enable smaller particles to be accessible.
Okay, good. Thank you. Kay has a question.
Yeah, thank you so much for the great talk. I was curious when you had your 100 kV samples, you said it was much harder to go to high mag. I'm wondering, was that just beam damage or was it a statistics thing, or what was the difficulty in going to high mag there?
The difficulty was practical. So I've never used a microscope in this range. And you have to calibrate. We use a cryo-EM software, which is the software we use to automate the experiment. So with these high magnifications, the fluorescent screen, you don't see much on it. And doing all the calibrations was challenging. In the end, there was also something related to the microscope configuration itself, because switching between it has mag ranges up to certain magnifications. The lenses work in a particular optical, design, so to say, configuration. Then going to the higher range, they switch. The kind of the strongly excited and not so strongly excited lenses, they switch the configuration, and there is a big shift between these ranges. And in the end, I had to go. I wanted to collect at 600,000, but there was such a big shift that I couldn't actually make the experiment work. So I used 500,000, so one magnification lower than that. It was just such small practical details which kind of made things more difficult. And in the end, this data set, because this was maybe a few days before the camera had to be mounted, it had already some parasitic remaining beam tilt, so about 1 millirad beam tilt, which is compensated in software, but it's better not to have it there. So it was just practical, practical small details. If I have a few months like that, I would probably work them all out, of course, with the help with Joe and with aligning the basic microscope configuration. But yeah, it was a bit time limited this time.
Good.
Got it. Thanks so much.
Thank you. So the one question for those who want to do tomography with 200, 100 kilovolt, what kinds of considerations you would recommend?
Well, I would say go to a 300 machine. So it only makes sense to do tomography if your sample is thin. So you saw the plot here. Consider what the tilt angle range would be, and if you do plus minus 60, the sample thickness is doubled at the 60-degree tilt angle. So if you want to collect at 200, you either use a limited tilt range or yeah, I don't know. Depends on the type of sample. If it's a cellular sample and you want to do tomography on it, definitely just go to 300 kV machine. I don't think it makes much sense to. I mean, you can do it. Nobody will stop you, and you would get something. But I haven't seen any kind of team 200 amazing results coming out from tomography at lower voltages thus far. Maybe there are. Yeah, that's what I think.
So I have a question. When you operate the cold field emission gun, how long can you operate without doing anything to keep it clean?
It's between 6 and 8 hours. I typically do the flushing of the gun when the cryogen refuel cycle starts. So the microscope refuels the nitrogen every 8 hours or so, and then just do the flushing of the field emission gun at the same time to avoid interruptions of the acquisition in between.
Mm-hmm.
And it drops the intensity. The intensity drops by about 10% typically in this. Depends on the microscope. I guess there are variations between different guns, but that's the typical.
I see. And then in this ARM2 microscope for the 200 kilovolt, can the sample be tilted?
No.
Okay.
That's the limitation. I didn't mention that.
Yeah.
That's the limitation. You cannot do tilting. This means no tomography, no micro-ED, no tilted sample acquisition for single particles. So if you have a very severe preferred orientation, one of the remedies is to collect data with the tilted sample. And for those, you would have to go to a machine that allows tilting. Or there is another option which you haven't explored, is there are this kind of corrugated grid. So you can take a grid and then use kind of a die and what is that called? Basically kind of stamp it, and then it makes the grid zigzag up and down, and then you get different areas of the grid have pre-tilt. So that's kind of a practical solution which I haven't explored. It may enable actually tilted data acquisition on a non-tilting stage.
Yeah, I think that's what one used to do on the two-dimensional crystal, I guess, by bending the grid. Okay, good. Well, it has been an exciting lecture you kick off this morning, and congratulations for this excellent work, Rado. Keep going.
Thank you.
Okay, good. Thank you. Let me pass it back to Bob to introduce our next speaker.
Thanks very much. Well, yeah, Rado, that was terrific and very useful for our class who's thinking about all these things about which voltage to use and so on, what the resolution limits are. So thank you for that very stimulating talk. Now I'd like to move on to the material science part. I'll do the introductions this time because I've known Eric Stach, our speaker, he points out, for over 30 years now. And it's a great pleasure to invite him to give our talk. He used to be one of the new generation of electron microscopists when I was a little bit younger. But now he's, of course, mid-career and has just stepped down from being president of the Materials Research Society. So he's long been involved with in situ microscopy and many practical things. And he's even managed to juggle a great research career with starting a company, making in situ holders and so on, Hummingbird, which I believe he's now moving on to even better things. Anyway, he might talk a little bit about that. But anyway, I'm very pleased to introduce Eric Stach to us today to give the material science talk. Eric, please.
Thank you, Bob. It's a pleasure to be called mid-career. I don't feel that that's accurate anymore, but it's nice to hear it. I'm going to go ahead and work on my Zoom sharing. So I have to do a couple of things here. I get it right. I was all prepared for this, but one second. Share. Desktop to share. How are we doing? Seeing a screen?
I presume it's full screen.
Yeah. Everything all right?
Looks great.
Yes.
Okay. Well, it's a pleasure to have the opportunity to present some of the recent work from our group. This is an area of research that I've been involved with for almost 10 years or more, looking at ways of trying to characterize catalyst systems in reactive environments using both electron microscopy as well as X-ray synchrotron-based methods. The work I'll show here is a result of two graduate students in my group, Alexander Fouché, who's presently at the Oak Ridge National Laboratory as a staff scientist, and also George Hollier, who is in his 4th year as a PhD student with me. This work has been kind of developed over the whole time with Anatoly Frankel as my key partner for the synchrotron-based X-ray work, as well as his former student, Nicholas Morcella. Ryan Tapero is our key collaborator at the NSLS2, and then a lot of the synthetic work for the nanoparticles comes from Christopher Murray's group here at the University of Pennsylvania. This work that I'm showing was largely supported by an Energy Frontier Research Center effort that just ended called IMask, and thus was DOE support. So what I want to do is talk about a set of ideas that we developed back when I was working at the Center for Functional Nanomaterials at Brookhaven National Laboratory. That's this building here, and the electron microscopy group was in the back corner of it, facing the brand new NSLS2 as it was being built at the time, and again, across the street from the original National Synchrotron Light Source NSLS1. The idea was basically because of the close proximity of the light sources as well as the electron microscopy capabilities, we wondered if there were ways to think about leveraging those two distinct capabilities to better understand how catalysts change in reactive environments, recognizing that synchrotrons have a tremendous amount of ability to characterize things on a global average scale with a great deal of quantification as well as just interrogation through a wide range of different methods, but they do lack the resolution that electron microscopy has. So that was kind of the way we were thinking about this. So if you think about characterizing the structure of a material, we need to take advantage of a variety of different experimental approaches to have a full understanding of both the structure in terms of crystallographic structure as well as electronic structure. And in the field of material science, what we generally do is take a number of different experimental approaches to learn something about our sample, and then think about how they come together to give an overall picture. There's a paper that was done by my colleague at Brookhaven, Simon Village, he's also at Columbia, kind of noting that really what we want to have in the material system is some way to take advantage of a wide range of experiment and utilize computation as a way to get towards an overall structure for looking at a given system. Another really frequent approach, especially in the X-ray synchrotron-based community, is to use multimodal approaches. Unlike an electron microscope, there's lots of space around the sample, so it's possible to put in a wide range of different detectors, and that allows you to look at a variety of different measurements at the same time, and that can be particularly useful when trying to do operando or in situ experiments where you're looking at changes to the sample in real time. You can then take advantage of a range of different approaches to learn more about your sample. One of the key challenges with that, though, is that X-rays are limited in their resolution. The very best X-ray microscope will have on the order of a 5 nanometer resolution, and that's nowhere near the capability of what the transmission electron microscope can provide. And so there is a real need, I would argue, for electron microscopy to be part of the overall characterization emphasis in understanding these sorts of systems. So our approach to this was to utilize microreactors. This is a technology that's been developed, over the past 20-some years to allow us to provide high temperatures as well as atmospheric pressures of gases to samples. This is done by creating microfabricated windows out of usually silicon nitride, embedding heating coils on those using microfabrication. And what this allows you to do is to basically create a little small catalytic reactor that can reside within the electron microscope. A key realization is that these same systems, if you have a synchrotron with appropriate focusing optics, you can focus the X-rays down so that they also fit within that same window size, allowing you to use the same systems both in the electron microscope and walking it across the street there to look using microfocused X-rays. So that was really kind of the key idea behind all this to see what we could do with this approach. We utilized some systems that we developed at the company Hummingbird Scientific. In this case, we build systems which again have heating elements embedded within them. They can go to 1,000 degrees Celsius easily. We have methodologies for measuring the temperature and making sure that it's uniform across the sample. Also, when we built the system, we made sure that it was a good catalytic reactor, meaning it's fully bakeable and stainless welded so that you end up having a good overall gas handling capability. And in the case of this system, we're able to provide up to 3 gases as well as a nitrogen purge so that you can go from, say, oxidative environments to reductive environments. They also include residual gas analysis, and that's something to, again, help you understand how the process is occurring. And again, in terms of disclosure, I was a co-founder of this company. We also can build these systems so that they have different implementations for different X-ray end stations, and that allows you to then change how the cell parameters may be manipulated in order to get certain types of data out. And this set of data that I'll show today, we instead use the exact same cell geometry. And what this means is it means that as we're looking at things in the X-ray system as well as in the electron microscope system, we're correlating things almost exactly because we have the same reactor design, the same flow conditions, and the same temperatures. So everything's more or less the same. And what that allows us to do is to really kind of combine both of these approaches, right, where we do multiple techniques at the same time during a reaction, not at the same time, but it's in the same reaction condition, using both the X-rays and the electrons. And that really allows us to kind of combine all these things together. And in the course of the past 10 years or more, we've demonstrated the ability to do X-ray absorption spectroscopy, infrared microscopy, and the full range of environmental electron microscopy techniques, including imaging, diffraction, spectroscopy using this approach. So with that background, I will kind of now go through and just show two quick papers where we use this to look at bimetallic systems to understand how reduction and oxidation lead to structural changes within specific arrangements. So the first one I'll talk about is cobalt platinum system. Platinum is known to be the optimal catalyst for electrocatalysis. It has just the right amount of both binding to allow the reactions to occur, but also the ease of the reactants to come off of the metal surface. However, platinum is extraordinarily expensive, and so much of the field over many years has been trying to find different alloying systems to help reduce the overall cost, but also to do things like allow enhanced stability to the catalysts and things like enhanced reactivity and overall selectivity as well. And in this case, there was a paper done by my colleague, Christopher Murray, and his former student, Jennifer Lee. She's at UC Merced now, where they were looking at these platinum nanocrystals and using that to understand how to improve the so-called oxidation reduction reaction. And so we were interested in building upon that to understand the nature of the particles as they went through both oxidative and reductive environments. So using the electron microscope, this is just simple imaging happening before the reaction. You actually can see that these things have a very faint contrast on the outer surface, and that's because the cobalt, which is much less heavy, is scattering less strongly. And so we actually see that upon synthesis, these have a relative cobalt segregation at the outer surface. And that was true for really all the particles that we were looking at before the reactivity occurred. So we go and use the X-ray fluorescent microscope beamline at Brookhaven National Laboratory. This beamline has the ability to focus the probe down to around a couple microns. We tended to use around a 10 micron focus. What that allows us to do is to look as we go through and do things like reduce the sample in hydrogen at 500 degrees Celsius, and they'll go back and oxidize it again at 300 degrees Celsius. We see changes in the near edge structure, which again are consistent with changes in the electronic structure of the cobalt. We can actually see that as we increase the temperature in hydrogen, we're able to get more and more overall reduction of the catalyst, bringing it towards a more metallic state. But we do find that when we compare that with pure metallic cobalt, it hasn't reached the whole way. So that indicates that even in these very aggressive environments of high temperatures with pure hydrogen, we're unable to reduce these catalysts completely to the metallic state. And this is, again, consistent with the so-called extended absorption fine structure. What you do with extended absorption fine structure is you look at the information past the edge, and you go through a series of fits that you can use to find out bond length, for example. And really what we were able to see with this is that on average, as we go through and do a reduction, we get to something that has both metallic cobalt as well as a CO oxygen, cobalt oxide. But if we go through and then look at the oxidation thereafter, it changes to a very different form, which is the CO304. So again, these are the sorts of informations that one can obtain overall using X-ray absorption, which samples a large number of particles in the reaction environment. One more, here we go. Similarly, we can complement that with electron energy loss spectroscopy. It's a consistent picture. We see that we do have both metallic cobalt and oxidized cobalt oxide samples throughout. So depending upon which particles we look at, we will see either purely metallic or some oxidized sample. We often will see there's this pre-peak that's present even after 500 degrees in the reductive environments. That's this little bit of red here before the edge. That's actually indicating that there's still a small amount of oxygen present. And overall, there's really a kind of an ability to use the electron energy loss spectroscopy to discriminate between individual particles and show how they make up the overall average that's observed by the X-ray absorption. If we want, we can also do energy dispersive X-ray spectroscopy. We again see that in this case, after reduction, the cobalt and platinum remain broadly mixed, and there's an oxygen present throughout the entire structure. If we look at images, right, we can go through and see how the particles change as a function of both reduction and oxidation. You see that in the initial stage, there's a relatively narrow size distribution, but upon reduction, we begin to see both coalescence. That's evident here from these two particles that have come together and sintered, as well as some evidence for Ostwald ripening, meaning that we have small particles and larger particles where the large particles are stealing some of the atoms from the smaller particles to reduce the overall surface to volume energy. Again, if we look at the oxidation, we see further coalescence and ripening occurring. If we also look at this in atomic levels of resolution, we see that in the initial stages, that cobalt that's segregated to the outer surface forms in the CO2-P3 phase. But if we go through and reduce that, we see an homogenization of the sample where the platinum and the cobalt are more generally agglomerated. But if we go through and do the reoxidation, we actually see a very coarse oxide layer on the outer surface. Again, this is all taken at 300 degrees C in an oxidative environment. And so this feature here is again consistent with the CO3O4 that was observed in the X-ray spectroscopy. If we look at this at lower levels of magnification, we actually can see that some of the sintering that occurs is not just from standard particle migration and Ostwald ripening, but also there's a degradation of the support. In this case, it's a carbon support. And as the support degrades, you begin to see particles come closer to each other. And again, that's something that can lead to the sintering. And this is the sort of thing that would not be evident by just using the X-ray absorption. You would not have any knowledge as to the origin of changes in particle size. Furthermore, if we look at this after the oxidation stage, we see again cobalt segregation to the CO3O4, again evidenced in the energy dispersive X-ray spectroscopy. And also we find by looking at the electron energy loss spectroscopy, it was a bit of a surprise, but it turned out that the larger particles were the ones that were easier to reoxidize. And those are the ones that also have larger regions of 3O4 segregation. So again, all of this is interesting because we're actually able to see that it's consistent throughout. It's difficult to fully reduce the cobalt in these cobalt platinum particles. And again, as you expose them to an oxygen environment, they undergo a transition to a different outer surface oxide phase. So this is a paper here, if you're interested, kind of describing all of this. And I think a main message to take away from this is that if you were able to just use, say, X-ray spectroscopy to understand what was happening, you would have a harder time seeing the origins of why you're forming the different phases, their outer surface segregation, for example, or any of the heterogeneity that's leading to the overall changes in the structure during the different environments. I'll show another example of this, again, from the same work. The same larger collaboration in this EFRC. We actually collaborated with a different EFRC down in Delaware on this, looking at nickel copper 3, which is again a different catalyst used for biomass conversion. In this paper, again, Chris Murray's group showed that this was an effective catalyst for changing from this form to this form of a biomass, hydroxymethylfurfural to dimethylfurfuran. This is one of the steps that you need to make in order to take biomass and convert it into ethanol, and a key step in the process. So again, we went through and did characterization of the samples beforehand. It turned out that there was actually two types of samples present during the synthesis. In one case, we had core shell particles, where you had a copper core and a nickel shell on the outside, but also in some cases, the particles were hollow. And again, in this case, it was observed that there was copper segregated to the outer surface in these hollow particles. So by doing a large amount of just imaging, we can get a reasonable understanding of the relative percentages of the different types of particles. And so this is actually important for catalysis because if you have heterogeneous systems on the way in, it makes it even more challenging to understand which of the types of particles are the ones most important for undergoing the catalytic reactions and driving the overall selectivity and activity. So again, we can use eels, right? It turns out that eels, this is ex situ, so before any reduction or oxidation. The eels is again consistent with this. The nickel is oxidized in more or less the same state in both particles. But if you look at the copper in the hollow particles, there's a different near edge structure that's again consistent with it being a more oxidized structure, closer to the CuO oxide. And that's again evident in the presence of the sharp peak. Whereas if you have a more metallic structure, that L3 peak is reduced. So a key point is if you have changes in both the oxidation state as the structural state for these catalysts, if you're using an average technique like the X-ray absorption, it can be hard then to learn which of the different particles are the ones involved in the reaction. So in this case, we went again back to the XFM beamline at Brookhaven. And interestingly, we found that if we looked at relatively high magnifications for that instrument, there were different regions of the sample that showed differences in the relative a number of particles that had copper and nickel. And so it indicates that not only was there heterogeneity in the two types of particles, but some of them tended to cluster together to give, say, more copper in one region or more nickel in another. And you can see this variety present by looking at the different spots. You're looking at the copper here. This is the K edge, and you see changes in the near edge structure, again, indicating differences in the oxidation state. Whereas again, just like in the eels, the nickel was more or less the same. So we can go through and again, do the reactions, holding them at high temperatures. We find that in doing so, the nickel does not change dramatically during reduction, but of course, as expected, the copper does become more metallic, but not all the way towards the state that's present for the copper metal. In other work, people have indicated that if you reduce nickel-copper alloys, you're going to end up basically creating a structure like this, where nickel and the copper oxide will reduce to nickel oxide and copper. And so we'll talk a little bit more as we go further about that reaction. If we go back and reoxidize it, we find that we actually get a copper 2 state. And again, that's consistent both in the near edge, where you have a strong pre-peak in the electron energy loss spectroscopy, but also in the X-ray absorption spectroscopy, indicating again that these are showing the same types of information here in the L edge and here in the K edge. And furthermore, if you look at the extended X-ray absorption signals, we again see the presence of the Cu2 oxide after the reoxidation. So if we go back now and image, we find that the core shell structures are removed during high exposures in hydrogen, and you end up instead having a copper-nickel mixed phase that often has a thin nickel oxide surface layer. So again, that's consistent with this equation down below. With time, there's a progressive and slow disintegration of the hollow structures. They don't completely disintegrate, but they begin to collapse upon each other and form solid spheres, again, with a nickel oxide on the outer surface. And that's evident within the images as well as the electron energy loss, looking at both the copper and nickel edges from sample to sample. Furthermore, if we look at this with STEM imaging and EDS, we can see that as you go to high temperatures, we begin to have these things break apart. There's segregation of the nickel oxide on the outer surface, again, due to changes in how the hydrogen is basically reducing the copper and leading to a segregation of the nickel oxide overall. And then really, if we go and look in oxygen, we have to go to very high temperatures. And if we do that, we again see that there's a collapse of these structures, the segregation of the copper and nickel. And all this is, again, consistent with the coordination number and oxidation state observed by the XFs. So just like within the previous example, I think the main takeaway from this is that when you're trying to understand how these catalysts change in reactive environments, having more looks at different levels of resolution, using both electron microscopy techniques and X-ray absorption techniques adds real value to characterizing things. Just to kind of finalize, we've done a number of different things over the years with this. We've demonstrated X-ray absorption spectroscopy and IR microscopy using beamlines. We can do electron PDF, small angle electron scattering, like this is just kind of like small angle X-ray scattering and imaging. We can understand things about the short range and long range order using both XAFs and XSELFs. That's actually something we're working on quite actively at the moment. Charge exchange with the support, changes the core shell structure. We have one paper looking specifically using the IR microscopy to understand how absorbates are affected by changes in catalyst structure. You can understand things about the support using Raman spectroscopy. And again, using chromatography and RGA, you can actually correlate these things specifically to changes in the reactions, specifically looking at things like how absorbance are related to changes here. Maybe one final thing that's relatively new and I think quite exciting is that working again with Hummingbird Scientific, we've developed a new holder that will allow us to go extremely high pressures. In this case, we're able to demonstrate up to 450 degrees Celsius, 20 bars, that's 20 times atmosphere, in this case of CO2 to hydrogen. We use the environmental TEM at Brookhaven because if these cells break in our normal instrument here at Penn, we would lose the cold field emission source because of the gases flowing up towards the gun. But in the case of the environmental TEM at Brookhaven and other environmental TEMs, there's additional differential pumping, which allows you to kind of keep these things safe, you might say. And with this, in the latest work from December of this past year, you can see very nice images at 20 bar and 450 degrees C for a platinum supported on Syria. And if you go and look deeper, there's clear images of 2.74 angstroms being resolved in the Syria 200, again at 20 bar and 450 degrees C. We know we have 20 bar because if we look in the electron energy loss spectra, it's just one big hump of scatter. But the imaging here using the zero loss filtering allows us to recover atomic levels of resolution. Furthermore, we can do electron diffraction at these high pressures. And again, this is a kind of a paper we're pulling together based upon the things we saw at these ultimately high pressures that are industrially relevant. So again, lots of different papers on this. Feel free to look up my name as well as Anatoly Frangel, and you can see the different things we've done, like correlate gas chemistries with different types of hydride and carbide formation, relate these phases to the presence of oligomers on the surface, understand a range of different approaches for doing this. And so my summary would be that this microreactor design allows us to do these correlations, including at industrially relevant pressures and temperatures. You can see that this gives rich insight into the changes that can occur, and that in these bimetallic systems, the changes are not singular, like only one thing happens, but rather many things happen. And I would argue that requiring that these sorts of types of images and spectra allow you to have a deeper sense as to what's going on at both the nanometer as well as the metascale for these systems. And so that's what I wanted to share with you today. And I guess I'll take any questions. And I see there's some. Bob, do you read them or do I read them?
I will try, but thank you. You can read them while I'm going to the guests. So thank you very much, Eric. And I have some of my own questions as well. But let's start with Patricia from Cape Town. And what's the idea why the nickel would not reduce completely?
Yeah, I don't know that I have an answer as to why. I can just say that that's the observation we have, even under these really aggressive temperatures and pressures. And so I think that in some extent, if we go, how do I go back up? 1 second. If we look at, let's see, let me get rid of this here. All right. Yeah, so we're basically saying, we got pure hydrogen at 400 degrees C, right? We see that there's this change where the copper and the nickel mix well, but they form this nickel oxide on the outer surface. So it appears as though we start off with this pure nickel. We had the copper oxide present in the first case. And what's happening instead is that the oxygen is preferentially moving towards the nickel and away from the copper in this environment. So it's as though the existence of the copper oxide at the beginning provides the source of oxygen for the nickel oxide that forms. And that's just the observation we have.
But you don't reduce it completely, though.
No, just can't. No, and one of the things about this is we probably could have gone to maybe higher pressures. We could have gone to higher, longer times. But to some extent, with beamline experiments, you tend to be limited by the overall amount of time that you have, to run the experiments.
Okay, good. Nicholas Marchese has asked about the electron beam, and does it cause any reaction itself, or?
There are times when you will see that. In these cases, we did not notice that in particular on these samples. And generally what we will do is that we will run the reaction condition for some extended period of time, like the 30 minutes at 200 degrees C. We may image one area, but of course, we'll always go and look at other areas to see if there's changes that are different, right? If you see something different, then that indicates that there's strong electron beam effects. That's kind of a standard mode for electron microscopy, is that if you do look at one region during a reaction, it's always wise to cool the sample down, take a look later in a different region to make sure that the morphology is consistent in a region where the electron beam has not been. And we did not see that in this case, that there was any electron beam effects.
What sort of currents are you using? What sort of doses?
It depends upon whether we're doing the STEM imaging, the EELs, or the EDS. There's a variety of different configurations that we use. This was done in the aberration corrected NEOARM instrument, 200 kV. So there's some good dose that is needed to do the EDS and the EELs. We use, it's called 6C spot size, 5 or whatever. No, spot size 6, sorry, in that. So there's, you can go through. I'd have to go and remember the exact correlation with that, with the current.
Yeah, our experience has been that the electron beam really does ionize the gases, and the reactions are a lot different. So,
well, and in this case, what I want to indicate, right, is that when we look at these things, we don't tend to image during the reaction, right? We'll go through and run the reaction so that we're waiting to see what happens, and then you can cool the sample if you want to check, or you can do it at high temperatures. But at that point, after 30 minutes, say at 200 degrees C, you see what you see. And the short exposure for the imaging doesn't tend to do that much more.
Okay,
makes sense?
Yeah. Nestor has a question.
Hey, Nestor.
It's about the EELs work. You can read the question. It's about the oxygen K overlapping with the cobalt L. I think you had labeled as cobalt K, but it's cobalt L shell. Do you have any problems? And is there do you have your silicon nitride window, which is completely oxygen free?
Let me read. That's too many questions at once. Let me check here. That is a good question. So there is a possibility of having oxygen on the silicon nitride for sure, Nestor. But I will say that if you look at, in this case with the cobalt platinum, we actually will see, let me see if I can go back up. I have a new keyboard and it doesn't have a page up. So let me see, how do I do this? Cobalt platinum. To some extent, Nestor, this is more or less one of the nice things about having the dual information, is that you can see that even in the case of the X-ray data, right, which has no electrons in it at all, right, you see there's the same formation of the cobalt oxide. But if we go through and look at things like the EDS and the EELs, where am I? Here's just imaging, right? So again, I think there's no question that you have the formation of this oxide shell here. And so I think that's, again, consistent with the EELs. So
I want to follow up on Nestor's question, because you're looking at fine structure here, and I'm wondering what the effect of the windows is on the resolution of the EELs, both at the L shell, which is about 600 or 700 electron volts. But also, if you were to compare the X-ray data, which is looking at the K shell, which is a much higher energy, the prospect of obtaining with these new energy loss spectrometers, the K shell data with the membrane.
And so, in fact, we already have data. George has gone up to use the instrument at Lehigh, where they have one of the new CEOS spectrometers on it. As well as, I think, a Dectris camera at the end. And we are specifically working with Masashi and Anatoly to push more on the XELFs data at high energy losses. So that's something that we're actively working on.
Good. And like Nestor, another former MSA president, says he's hoping that you put the high pressure paper in for microscopy and microanalysis. And John Mansfield.
Well, thank you very much, John. We've been putting these in MRS Communications as president of the Materials Research Society last year. I had a goal to publish in each of the MRS journals. I made 3 of the 4. So that's where we publish it in MRS Communications.
I think Kate has a question. Kate Reedy.
Yeah, thanks, Eric. That was an excellent talk as always. I was curious, at some stage, I think you mentioned like a carbon support.
Yes.
Or support of these nanoparticles. Did
Yep.
you ever see any carbide formation when you went up to these high temperatures?
No, we did not see any carbide formation in these particular cases. Yeah.
Okay, cool. And these nanoparticles were created by like chemical synthesis too?
Yeah, so this is Chris Murray's lab. So Chris Murray, he does the colloidal synthesis method. You know, he was Wendy's student back in the day when that Nobel Prize paper was produced. So they're very good at making very uniform colloidal nanocrystals.
Do they ever have any contamination on their, not contamination, but like ligands on their surface?
Sure, absolutely. Yep. And we tend to do some plasma cleaning to get rid of that. Working actually, Jennifer Lee, the student who's on a couple of those papers, we worked pretty carefully to try to figure out how to get rid of all that stuff.
Okay. Yeah. Thanks.
Yeah.
So Eric, for those people who don't have a synchrotron in their backyard,
yep,
and just have an electron microscope, you know, how would they best do these experiments then, in your estimation?
So what I would say is that the electron microscopy, because it has the ability to do imaging, diffraction, and spectroscopy, 2 types of spectroscopy, maybe even 3 with the XL, so if you consider it separate, is a pretty versatile tool for doing a lot of this stuff. I think the advantage to adding the X-rays on top of it is it tends to give you higher confidence in making statements that are from small statistics, right? It's hard to do tons and tons of electron energy loss spectroscopy because of the time it takes to do so. And if you have, you know, the ability to have the X-ray data, it gives you a sense that you're getting more information that's consistent across that. I think also in some cases, like the IR microscopy, that's one we've only done 1 experiment for, but that was a really nice and fun experiment because we were able to do things like use the diffraction capability as well as imaging capabilities to understand something about the structures of, in that case, platinum carbide and platinum hydride. Palladium carbide, palladium hydride, and correlate that with observations of different chemicals on the surface of the material. So that was kind of a fun one.
Good. Let's see. I'm not sure if Colin is still online. I know he's at Arizona State. I'm not sure if Colin has any questions. And or Kate.
She asked hers.
Yeah, I was also curious. These really high pressure cells seem incredible. What did you have to do, or what was the secret to getting atomic resolution when you have the 20 bar there?
It was a surprise, and actually, I will say that I know for a fact that so we did our first paper was on using STEM only, not high resolution TEM. And one of the reviewers said, "Well, why didn't you do zero loss filtering in high resolution TM?" And it was actually Stig Helveg. I talked to him later, but he was right. And so it actually the zero loss filtering does help quite a bit with that. We use very narrow spacers, so there's not a lot of. We don't have like a 1 micron gas level. We got 100 nanometer. They bow somewhat like always, but we are using extremely small windows. So in the case of the normal windows, we use 250 micron by 50 micron in the hummingbird cells, and that's the 50 microns is to limit the bowing in one direction. In this case, we're using extremely small windows, 2 of which overlap, right? So they don't have too much bending outwards, if you know what I mean. Yeah,
that's incredible.
So you have to actually. So before doing the experiments, there's a fair amount of work to do the catalyst deposition, and also to make sure that you do a little bit of SEM work to make sure that you have something on the windows before you go to the effort of doing it. Yeah, because it's a relatively small. I mean, it's like 2 microns by 2 microns. Imaging.
Wow. And then finding it when you put it into the
yeah.
That's incredible.
Yeah, you could hardly, if it's 2 microns by 2 microns, you couldn't even find it on the optical microscope with ease anyway. So, I'm also glad to hear that you put these things in an ETEM because of protection, and we've done that with our liquid cell as well because we're paranoid about these things breaking inside the machine. And
we only do liquid cell work on our F200 here. We no longer do it on the Neo arm due to one very expensive break.
It is indeed.
Yeah.
Okay.
But yeah, the ETEM, it turns out that the way, just is kind of, if you have just 2 seconds, the way we figured out what pressures we could go to is we actually took the membranes and poked a hole in them with the tweezer, right? And then stuck them in the microscope, ran the ETEM mode, and then just turned up the gas pressure on the bottle, right? And saw at, there's the gauge that sits right above right by the gun valve, right? And so we basically just kind of watched that until it hit 1500, which is the one where you go, ooh, that's kind of scary. And we were able to get up to 50 bar in that instrument without it crashing.
Wow.
And that's with consistent flow of 50 bar. So the thought is if you have windows that are intact, but then they break, then you know that that's happened and you can shut things off.
So let's go back to the ionization caused by the electron beam. So yeah, we have often done experiments whereby we turn the beam off or blanket completely to do the oxidation or the whatever it was, the reduction. And unfortunately, that seems to be necessary. So the taking in situ movies, it's a completely different thing when you got the beam on with the ionization effects. So you agree?
Yeah, I think that could definitely be the case. Yeah. It depends also on the amount of flux, right? If you're doing atomic resolution imaging, for example, right, you get the beam like janked down on your sample really hard, it'll definitely be more of a problem.
Yeah.
And of course, STEM imaging mode is better than high resolution TM in that regard too.
Yeah. But then you have we can't do the in situ. So
well, not as fast. Yeah.
Well, it's frame by frame.
Yep.
Yeah. So if there are no more questions, I think we can close the session off. I'd like to thank Eric very much for this great talk and pass it over to Wa for comments which he might have, please.
Okay, I think this is really fascinating. You can put such a high pressure in the electron microscope and such a high temperature. We have never done that in biological samples.
I would doubt it. Yeah.
I don't think we need to.
I don't think you need to. Yeah, but I mean, importantly, if if you go to an industrial reactor, they will operate at these pressures. So there has always been this question around pressure gap, and so it's important.
I'm just curious whether such a kind of setup can be done in a more modern microscope. In other words, you still use a F20, is that right? This Thermo Fisher microscope?
No, this was done on a Titan environmental TEM, so a dedicated environmental TEM.
Okay.
Yeah. So that was installed in 2009 at Brookhaven. So it's still operational there, and a good instrument.
Okay.
Yes, our Titan also, all ETEM 2010.
Yeah.
Same thing.
I remember.
Still working great.
Yep.
Okay, that sounds good.
But I would say that does indicate that this high pressure approach is going to be valuable at Stanford, Brookhaven, Copenhagen, Osaka. It's not the sort of thing that's going to make the company a lot of money. Relatively limited clientele.
We'll be excited to use it at Stanford.
Exactly. Yeah.
Okay, that's great. Okay, I think right now we can close the sessions formally, but then we're kind of rather stay up is at kind of 2:30 a.m. In Tokyo time. Thanks so much, Rado, staying up with us. And I'd like to open the room to any question from the remaining participants. Please type it on your Q&A.
I think it looks like we're set to go, Laura.
Okay, any questions?
Well, if Nesta has to leave, I think we should also. So thank you very much, everyone, for a great session. And we'll close off the recording now, please, and then we can carry on chatting.